introduce himself. So Christoph, if you would like to go right ahead.
Hi everyone, my name is Christoph Heubeck. I'm a professor of geology at Jena University, and I'm here the chair of what is it called? A general geology and earth history. And by training, I'm a sedimentologist and stratigrapher and sedimentary petrographer. And I used to have 6 years in the petroleum industry. And for the last 10 or 15 years, I have become somewhat of a specialist for the Barberton Greenstone Belt, in particular for the Moody's group there. And typically I have 1 or 2 field seasons a year in South Africa. And however, under these COVID restrictions, of course, that's not possible. And therefore, I'm really glad that I was invited here to participate here in the lockdown lectures. Enough of an introduction, or are there,
That is perfect, thank you. You can go right ahead.
Then let me share my screen, and you please tell me whether you actually, whether you see what I want you to see.
Yes.
Okay.
Perfect.
Yeah, presentation mode, and you can see and hear me?
Absolutely, you can go right ahead.
All right, so I was asked by Chris to talk about early life on land, and that's a tall order. Okay, so I tried to combine this by writing this title that you see here, New Insights from the Barberton Greenstone Belt, including evidence for early life on land. And so the structure of this talk, which will have 69 slides and will last for about 40 minutes or so, is pretty simple: intro and motivation. And then we have 3 lines of evidence, which I'd like to present to you. And then there's a final chapter on other news from the Barberton Greenstone Belt. And I should start out by just flatly stating that most of what I know about the Barberton Greenstone Belt, I know from my students who have been down there with me, and they have all done field work. There must be about 20 by now. I think the earliest started in 2007 or so. And currently I have the 2 PhD students that you see down here in the lower right. That is Sebastian Reimann and Dion Janse van Rensburg. We know very little about the early Earth, right? And we were, so in particular, we roughly know where the oldest rocks are and maybe even the oldest minerals, but we know really little about what really counts in life. We know nothing about continents, the atmosphere, the coasts, the oceans, climate, weather, radiation, volcanism, tides, meteorites, and so on. And all of you could probably come up with half a dozen of questions to each of those topics. And most importantly, we know very little about the origin and the early evolution of life. Life, which is up to now, at least, unique in the universe. Even though we by now know a couple of 1,000 planets, of which a couple of dozens appear to be habitable, but life is that big mystery and enigma. So in order to dive a bit, not into the origin of life, but how life, which undoubtedly originated and formed in aqueous environment, how life conquered land, I'll take you to the Barberton Greenstone Belt. And I think most of you are quite familiar, so I'll quickly go over these slides. We are here in some of these very oldest, well-preserved rocks on Earth. You see in the green rectangle on the timeline here, which goes from the left to the right, the age of the rocks exposed in the Barberton Greenstone Belt. They are between 3.57 and 3.22 billion years old and sit in the Paleo-Archean, if you want to subdivide the Archean any more than necessary. This was a dramatic time. As you see, planet Earth was ruled by bacteria. Most of life was anaerobic. Photosynthesis had been invented, probably, and was pumping oxygen into the water or into the atmosphere. However, most or all of that oxygen was absorbed by minerals that were ready to weather by oxidation. Think of all the pyrite, for example. Barberton is pretty unique. I think the Barberton Greenstone Belt you see here is, and I only realized that in the last few years, is unique not only for its geology, but also for its topography. It has a topographic range of 1,700 meters, the highest peak being south of Barberton in the middle of the Barberton Greenstone Belt, made of the mists with 1,900, and the lowest point probably in southernmost Kruger Park at around 250 meters of elevation. So a tremendous range of 1,700 meters, which is very special and unusual for rocks of that great age, because these greenstone belts, along with the Archean cratonal basements, are typically within the interior of continents, and these are typically flat because they are not much affected by plate tectonic processes. However, Barberton is different. Just in case we have one or two people among us here who are new to greenstone belts, here is an introductory slide. What is a greenstone belt? Greenstone belts are sedimentary and volcanic remnants of former surface sediments on volcanics that are deeply infolded between older, coeval, and younger plutonic and metamorphic rocks. And you see here in this very schematic diagram that shows, however, some similarity to Barberton, how this geometry has to be visualized. You see in the pale green, brown, and yellow colors the greenstone belt units, and surrounded by these various generations of plutons, some of which have moved as they cooled and therefore acquired a marginal fabric so that they look like gneisses. But they are really plutons that have acquired an outer shield or rind. They are called gneiss domes. And they are in turn intruded by younger granites that I show here in orange, and that typically take on a sheet-like geometry. And in southern Africa, as we all know, the cratonal basement that crops out and in Mpumalanga, in Kruger Park area, and Barberton and northernmost Swaziland is overlain by flat-lying cover rocks, which also have an immense age, but are completely unmetamorphosed and flat-lying. And then, of course, over to the east, in the case of Barberton, we have younger strata that are Triassic or Cretaceous in age. They are mostly in Mozambique. I don't have to explain this map. I think this is the map, which is now almost 40 years old, by Karl Anhauser and co-authors. And an oldie but goodie, I would say. By now we know much more about the age relationships. We probably would not subdivide the Unverwacht group into blue and green anymore. And we would have a higher number of units. But this is still, I think, to my knowledge, the only full color map that extends across both countries, South Africa and Eswatini. And you already see the basic stratigraphy, Moody's, Victory, and Unverwacht group. Here, a bit more detail. The stratigraphic sequence in the Barberton Greenstone Belt, the fill of sedimentary and volcanic units is immense. Look to the left. These are almost 16 kilometers of stratigraphic thickness. By now, it has been well established that this is true stratigraphic thickness and not tectonic repetition. And about 2/3 are made out of the volcanic, mostly volcanic rocks of the Unverwacht group, which probably represents some sort of oceanic plateau and takes up almost 18% of the Greenstone Belt's fill age, overlain by the Victory group, about 2 to 3 kilometers in thickness, and a mixed volcanic sedimentary unit, and then overlain by the Moody's group, which I will then concentrate on in the following. And the Moody's group is one of the oldest, well-preserved, quartz-rich sedimentary units on Earth. And notice that it's only about 5 million years long. This is what the Barberton Greenstone Belt looks like in the field. It looks like an anticline, but it's not. It's a syncline. It's, however, you're looking towards the plunge of the syncline. Here's the plunge, and that plunge is steeper than the slope, so that you have the topographic slope across that gorge, so that you have that impression, the false impression of this being an anticline. But no, it's the second quartzite MDQ2 of the Moody's group in the Stolzbrook syncline. Another steeply plunging syncline here, the Dysdale syncline, and well known to all of us, and Tony knows this, in particular knows this area as well as I do, or probably better. He has shown me quite a number of things. And in general, and you have seen this from the geological map already, the strata in the Greenstone Belt are highly deformed, steeply plunging and giving rise to a special nomenclature of canoe synclines and mushroom anticlines. But you notice the question mark, because it is by no means established that the steep plunge of the synclines that we often see where they bend around or where they end actually flattens along strike. So this is just to the end, the final slide of the introduction, because the Greenstone Belt is so deformed, and geochronological and structural data are deficient, I would say. A large number of tectonic models have been proposed what the Greenstone Belt actually represents. And pretty much every tectonic setting that you can imagine has been proposed. The uppermost row represents plate tectonic and therefore more or less shallow models, and you see the names associated with them. I redrew these diagrams from these publications and tried to give them a consistent color code, but didn't quite achieve that setting. The second row is an exercise in contrast, and it shows you the deep origin of and the deep formation of greenstone belts, left in a conceptual sketch by Karl Anhauser, and right in the drip model of Martin van Kranendonk. And the lowermost row represents cross-sections by authors who are sitting the fence and can't really decide. They use elements of both shallow and maybe plate tectonic terms, some of them, and deep tectonics. So I was among that group for a long time as well. You see to the left a cross-section resulting from my PhD thesis. And I think I'm even a co-author on that middle diagram here. And Alex Kisters from Stellenbosch here to the lower right subscribes to an extensional origin, which then sagged. So plate tectonics and deep lava lamp tectonics, you may call the middle row, and then these hybrid or intermediate models. But enough of that introduction and enough of tectonics. Let's look at life. And I'll start with, I think, 4 slides on marine life, because that is in the Onverbach group. Here's a field slide of the Buck Reef Chert, the lowermost unit of the Kromberg Formation, K1C, the Buck Reef Chert, 300 meters thick, one of the thickest or the thickest Archean chert unit, and has been mapped and studied by quite a number of authors. A typical rock type is laminated and bedded and sometimes cross-bedded chert, but the chert is a silicified. This can be a wide variety of mostly sedimentary rocks, of siltstones and sandstones, even conglomerates that are completely silicified. Here we are apparently looking at some laminated, fine-grained sedimentary rock that is stratified in light and dark stripes, and the dark is due to the kerogen. And when you do hundreds of thin sections and study them carefully, you will find some of these forms, I would say. They are kerogenous, consist of kerogen, of organic matter, that have been interpreted as being some of the earliest life forms, as long as you associate life with physical and morphological evidence, that is so-called body fossils. So see the my condition up in the upper right. These are some of the oldest body fossils on Earth. Any older fossils would be molecular or chemical in nature. You wouldn't really have any shape. And you see they are globular, like in the right and the center, and some of them are filamentous in the lower right, and many others are spindle-shaped. And we have no idea what they might be or how they lived. And the age here would be about 3570, or no, 3300, sorry. So Missouri chert and other cherts of the Menden Formation in particular are quite rich of former organic material, some of which show these particular ovoid or globular or filamentous forms. However, I would like to focus today on evidence that tells us that life gradually moved out of the oceans and at least into the tidal zone. What on earth could have caused life to do that? What is the advantage of moving away from the open ocean and into the shallow coastline where you endanger yourself to being desiccated or burned by UV radiation? Well, two advantages are, one, you have a firm substrate, and firm substrates allow to be colonized at a much higher cell density than planktonic and benthic environments, planktonic environments floating passively on the open oceans. I think the multiplication is a factor of 1,000 to a factor of 100,000 if you can grow on a firm substrate. Secondly, of course, is weathering from the continents, which are exposed to acid rain and where dissolving mafic minerals and feldspars are abundant, flushing everything that life needs, such as potassium, magnesium, iron, phosphorus, calcium, sodium, into the adjacent coastal strips. So food aplenty. And thirdly, while you are in shallow waters, you have direct access to the atmosphere, including its radiation. As long as you manage to protect yourself from the harmful high energy UV radiation, you have lots of lots of energy to be used for photosynthesis. So it does have advantages to go into the tidal zone. Let's look at the Moody's group here. Moody's group looks like this. Sebastian knows the area quite well. We are here in an area called Die Schweiz, Switzerland, but that is a field anecdote. The Moody's group is one of the oldest well-preserved quartzose sedimentary units on Earth. It is rather well exposed. It has up to 700 meters of relief, many places where you don't really want to fall down. Most strata dip subvertically, and Moody's group reaches up to 3,700 meters in thickness in the Saddleback syncline and also in the Eureka syncline. And it is traceable virtually throughout the entire Barberton Greenstone Belt, an area of about 40 times 100 kilometers. Moody's group sandstones are full of sedimentary structures. And you see the ripple marks, you see the faucets. Some of them are bidirectional in the upper right. You see how the tidal currents go to the left and to the right. There are mud cracks in the lower left, and there are these microbial mats that are laminated and stacked and apparently rhythmically bedded, which I will talk about a bit more in the following slides. We think we know fairly well what depositional environment Moody's strata were deposited. They cover a range from perhaps alluvial, but a lot of fluvial and coastal and deltaic and tidal sedimentary environments. The deepest I have seen would be pro-delta, so maybe 50 to 100 meters water depth. And the shallowest we have seen would be way deep into the mountain ranges, extremely coarse-grained debris flows and lahars and so-called bajadas, that is the products from rockfalls. But most of the Moody's group strata actually are, as I show here in the slide, representing coastal plains and deltas, and of course completely without vegetation. and probably under the subjected to high tidal range. My students and I have mapped and measured throughout the Moody's group for the last 15 years or so, and it's been great fun. Here you see Martin Hohmann standing on the second highest mountain in the Greenstone Belt, Chokolwa, and some of the stratigraphic sections he has measured here in the background. And if we look at his science, you see how these stratigraphic sections correlate with each other. You see from the background these faint blue, yellowish, and pink colors, that there isn't really too much variation. The horizontal scale here is about 15 kilometers, and the vertical scale 1,690, if you look at the number in the upper left. What is noticeable, and what I would like to direct your attention to, however, are the green units. And these are sandstones that are laminated by microbial mats. And if you look at the legend, which is not really legible down here in the lower right, you see that Martin distinguishes between a number of different phases. Let me briefly show you these microbial mats, because we interpret them to have that they dominated the intertidal and shallow subtidal zone. This is what they look like. They are very conspicuous and apparent in the field. It takes you no problem to discover them, but only in the Saddleback syncline, close to an intrusion, and there's a special relationship probably between the hydrothermal halo around that intrusion and the development and preservation of microbial mats, which is actually the topic of a paper that is currently in review, in which Sebastian is the lead author. So these microbial mats appear to have been pliable, and they are deformed by fluid escape structures. You see here a very nice flame structure. Some of these flame structures reach great thicknesses, up to 7 meters. Bulging and belching gases and liquids, and probably also some sand, into shallow sand volcanoes in the intertidal zone. The microbial mats form clusters and domes, and they appear to be nested, look a bit like cauliflowers. In cross-section, they form pinnacles, and these pinnacles appear to have been sturdy enough that they were not overly deflated when they were covered by the following layer of sandy sediment. And we imagine these tufts and pinnacles to have looked like the lowermost figure here, the lowermost photograph, which is obviously a recent photograph. Some of these microbial mats overgrow conglomerate clasts, in particular these clasts that are sticking out, like the one you see here in the center left, a feldspar porphyry clast, and you see how it's overlain by these dark green bands that actually thicken towards the top, and then also feel comfortable over here. And then you see that the relief has been smoothened out by the next sand layer before the next microbial mat was deposited. Some microbial mats are actually eroded, and where they are eroded, they form rigid chips in shallow and wide tidal channels. And we typically find desiccation cracks or mud cracks nearby. So this implies that they either desiccated themselves or that they stiffened by some other influence, possibly by carbonate precipitation within the microbial mat, or even by very early silicification. So none of this is really evidence for early life on land yet, and therefore I would like to present to you in the following 15 or 20 minutes, briefly, 3 lines of evidence that make us think that some of these microbial mats or microorganisms, even where they were not mat forming, actually were living in areas and now I need to phrase carefully, that were at least temporarily not covered by water, okay? Or not permanently wet, but just moist. So evidence number one would be sand blisters in the intertidal zone. And this refers to these black bands you see here in this cross-sectional shot of a field outcrop. And they consist of black chert, and they are interbedded and grade laterally, interbedded with and grade laterally into these microbial mats, kerogenous microbial mats, which I just discussed. When we take thin sections of those and look at those at higher magnification, they look internally like this. You see sand and sandstone above and below, and that the overlying sandstone is a bit finer grained than the underlying sandstone. It's also something we see typically. And then you see that these black chert bands are actually internally laminated, and by kerogenous material. We did Raman spectroscopy on those, and yes, they are kerogen. And where they are identified by these black arrows, you actually see downward protruding, downwardly bulging microbial mats. And for about a year or maybe 2 years, we thought that we had oriented our samples in the field or in the laboratory incorrectly, because we were used to microbial mats growing up in tufts and pinnacles and domes, not down as they do here. And only after a while, and after magnifying and thinking hard and long about this, we came to the and of course taking more thin sections and making sure that we would have firm control on their orientation throughout the preparation process. We figured out that these were something like stalactites. These were roof pendants. They grew from the top down, which in turn implies that this was a cavity. This was a cave in soft sediment that was partially or occasionally filled with water, but apparently as these microbial mats grew downward under the influence of gravity, apparently they occasionally were filled by air. When you crack open these microbial black cherts from these Moody's black chert bands and look at them and etch them and look at them under the SEM, you do see structures that look a bit like bacterial filaments. We need to follow up on that line of evidence a bit more. So we think we understand what these black chert bands mean. They are sand blisters, as they are common, for example, in some Tunisian tidal flats. This is southern Tunisia, close to the border to Libya. And this here is a close-up structure. And you see that these are apparently gas domes above or gas domes below microbial mats that bulged apparently when the microbial mats died and were coated by either aeolian or aqueously transported fine-grained sands, armoring the upper roof of the cavity. So we have, of course, we took many pictures, and you see the comparison of Moody's group structures to the left and modern structures to the right. So we think these sand blisters may be colonized by microbial mats, may be evidence that, that life was at least active in the intertidal strip outside the water at low tide, and protected itself from the harsh surface environments by a roof of several diameters of sand grains coated by extracellular polymeric substance, just like you see the gas dome in the lower right. Our second line of evidence is microbial sulfate reduction near gypsum concretions, because the Moody's group, to all of our surprise, actually does contain quite a number of gypsum concretions And those are characteristic of the transitional phases between the fluvial and the tidal environment. In particular, in the Saddleback and in the Stolzburg syncline, but we have found these gypsum concretions, which are now mostly silicified and show up here in this outcrop photograph as white clusters virtually throughout the Greenstone Belt. Many of these gypsum concretions, as you see here, are hollow and have apparently where some substance has been leached out, and in other places they are just imprints, and you see some reminiscent radiant and bladed texture where from the sandstone the formerly existing crystals, whatever they were, have been leached out. We think they formed as follows. First, you had the precipitation and of gypsum, and then the gypsum nodule was partially either carbonated or silicified externally. Then you go and dissolve the remaining gypsum, for example, during periodic or prolonged flooding, and then you refill it with either sericite, which is essentially clay, metamorphic clay, or a renewed growth of silica or of dolomite. In thin section, the gypsum clusters look like this. Yeah, they show a euhedral crystallographic pattern and overgrow other sand grains. And you see, more importantly, that they deform this black stripe, which is a heavy mineral, a detrital heavy mineral layer enriched in chromite and pyrite, probably a lag during the high tide. And when you look at these, at the pyrite grains within the heavy mineral lag that is wrapping around the gypsum concretions, and you notice that the gypsum concretions apparently were there first, and then when the sediment compacted, they were already stiff and deformed the detrital mineral layer. But when you look at the pyrites in there, you see that they show isomorphic rims, but they're not metamorphic, okay? And that raised our curiosity. How do you put new pyrite rims on pre-existing pyrite grains? Well, by precipitation of sulfide, of HS, combining it with iron 2+. So we took some of these pyrite grains to an extremely impressive machine, which is an ion microprobe. So it shoots ion beams very focusedly on parts of crystals, and the resulting ion stream is then analyzed in a mass spectrometer, and you can figure out the isotopes. So these here are the results for you see one representative grain in the lower left, and the numbers displayed there are the differences in the heavy sulfur, that is the 34 sulfur. Sulfur typically has a mass of 32, but we are specifically examining the abundance of the 34 sulfur in this pyrite grain. And you see that all the numbers in that homogeneous idiomorphic rim are strongly negative, typically about -22, whereas those in the dirty and somewhat lighter detrital core are typically around 0. That is, these numbers around 0 is typical for hydrothermal origin of a detrital pyrite grain, nothing special. But the negative numbers in the overgrowth rim are a clear indication of a fractionation that fractionates against the heavy sulfur, trying to precipitate new pyrite growth around pre-existing detrital grains that has been depleted in heavy sulfur. And that in turn is an indication of microbial sulfate reduction. So you see in the first bullet point here, we take the overgrowth of 34 sulfur depleted sulfur in the pyrite rim as an indication that light sulfur was in the water. How do you generate the light sulfide? Well, you take sulfate from the gypsum concretions, combine it with organic matter to make sulfide and carbonate. And in the next line, you see how I split this up in chemical writing. Calcium and sulfate ion plus organic matter, CHOH, makes sulfide, HS minus, and carbonate, CO3 double minus. And that is a microbial process. The microbes try not to use the heavy sulfur and generate sulfide, which is a gas. You could call it flatulence, which then grabs the iron and precipitates as a solid. So to speak in very anthropomorphic terms, these pyrite rims are fossilized flatulence of microbes. Microbes that have been eaten, that have eaten the gypsum, if they had mouths, that have metabolized the gypsum. Where is the evidence for life on land? Well, the gypsum concretions evidently formed on land, but they are not live. And the microbes that metabolize the dissolving gypsum are evidence of life, but not necessarily on land because they exist in an aqueous environment. However, the combination of both makes us think that this microbial sulfate reduction occurred in the occasionally flooded parts of coastal floodplains which contained gypsum concretions. So we think this is microbial metabolism active in the groundwater of the coastal zone. My third evidence consists of different proportions or of carbon and nitrogen isotopes in fluvial environments compared to their tidal counterparts. And that has been the topic of a Nature Geoscience article led by Martin Hohmann. And we go to the Dicedale syncline to look at microbial mats that are not associated with any indications of tidal evidence, such as mud cracks and reversing currents and the geometry of estuaries or tidal flats, tidal channels, etc. The microbial mats that you see here are rather associated with conglomerates. And I even have a slab of that here. And you don't see the conglomerate, the coarse-grained grains very well, except for some of these granules. This is definitely evidence for very shallow water. The pictures you see here show that these microbial mats are regularly getting ripped up into mat fragments shown in the lower two lines of this composite picture. And we are sure that this is not pyrite or black chert or anything else, but we are sure that these filamentous structures actually do consist of kerogen because we looked at this with the Raman spectroscope. Here you see in red the graphs for kerogen and the characteristic G and D peaks for graphite and for disordered. And you see that in both thin section images, A and D, when you convert them into a Raman map, that they have abundant organic matter. When you then do isotopic analysis on this organic matter from the Dicedale syncline associated with these conglomerates, and in the absence of any evidence for tidal structures, you see that these data shown here in green have two distinctions compared with the microbial mats from the Saddleback syncline, where they occur in tidal environments. That is, in the carbon plot on the left, you see that they are less depleted in carbon, typically showing values around -22, whereas most other values from the tidal zone have values between -25 and -33 or 34. And they also have different nitrogen isotopes. That is, they have a higher percentage of the heavy 15 mass nitrogen. It would take too long to explain what the processes are that lead to the enrichment of heavy nitrogen in microbial mats, and several pathways are possible. However, we think that the isotopic distinction in two different isotopes in combination with the facies environment would be another evidence that life on land, that is in the fluvial environment or the upper estuarine reaches of tidal deltas, may have developed a slightly different metabolism and adapted to the very special and somewhat more harsher environmental conditions in these environments. I can skip that because it relates to a slightly different topic. I would like to finish with a couple of slides on what's been going on in the Barberton Greenstone Belt in other news. We have in the last 2 or 3 years compiled about 30 maps, I believe, from the Barberton Greenstone Belt and digitized them partially or completely, and then tried to merge them. We compiled data tables on all the structures and the deformation phases, all digital, all the faults, all the folds. You see some of them named here in the map, in this map. We remeasured and compiled from the literature as well as from our own measurements plunge data from these folds, and then made a contour map, which you see here in blue, and found out that the highest plunges, most of them are vertical, occur here in the lower left, in the southwestern part of the Greenstone Belt, whereas the fold plunges shallow out and become more or less horizontal in the central part of the Greenstone Belt. So they may re-steepen again in the northeastern Greenstone Belt, making the whole fold bundle of the Greenstone Belt somewhat of a U-shaped structure. We also measured the proportions of conglomerate clasts, A, B, and C axes, and compiled strain maps in order to find out to what degree and where the Barberton Greenstone Belt had been deformed by ductile deformation. And you see here the first of 2 maps. This one shows the magnitude of tectonic strain, all derived from Moody's conglomerate clasts. And we have about 82 data points in here. You see the map is nevertheless incomplete, and you derive from here essentially that the information that the magnitude of tectonic strain increases towards the Greenstone Belt margins, as you would expect, and that it is lowest in its interior. And perhaps more interestingly, the second map here shows the type of tectonic strain rather than the magnitude. And those who recall their structural geology lectures would be familiar with the diagram in the lower right, a Flynn diagram showing the difference between oblate strain in the lower right and prolate strain in the upper left. Oblate strain means pancake-shaped. Prolate strain means cigar-shaped. And you see here in the Greenstone Belt that there's a big difference. Most of the clasts in the northern Greenstone Belt, and some of you may be familiar with the famous outcrop at Etsy's Pass in the Eureka syncline near Sheba Siding, are typically oblate. They are pancake-shaped and show only a moderate elongation, whereas most of the Moody's conglomerate clasts in Eswatini, down in the southern Greenstone Belt, are definitely prolate. They are cigar-shaped. We do not quite know yet what this means, except that the Greenstone Belt must be asymmetric. Asymmetric in its final deformation phase, and has undergone different type of tectonic deformation along its southern margin than it has at its northern margin. And my last point is that a renewed focus of our attention has become the Onverwacht Andecline in our efforts to reconstruct the tectonic history of the Greenstone Belt, because the Onverwacht Anticline is this big oceanic platform which has nevertheless been folded tightly and essentially tilted vertically. And I think that fact and that evolution has not really been considered sufficiently in previous publications. If you take the Buckreef chert, which is shown here in a dashed white line, it has a 30 kilometer, and trace it throughout the Onverwacht Anticline. You can trace it for almost 30 kilometers. So that must have been the minimum diameter of the Onverwacht platform. It could have been much larger. And so a very simple reconstruction of that Onverwacht volcanic platform is shown here in the lower part of the diagram. Now take that cone or that volcanic plateau and tilt it 90 degrees and fold it like a napkin, and you will get this here. Yeah. Now remember the dimensions. This structure, as shown here, extends about 15 kilometers into the air, where it has been eroded, of course, and it extends for about 15 kilometers into the to depth, or at least it did so in the past. Whether it has now been all incorporated into younger plutons at depth, we don't really know until we shoot seismic and do good gravity and magnetics. So this is essentially all I wanted to show you for today. Early evidence for life on land. We could discuss now for a long time what on land means, and that would take us all the way into Gondwana and into the amphibians and reptilians and the colonization of deserts and high mountain ranges. But I think I would like to leave it at that. Maybe the last slide here shown here is just an update on ICDP, on the drilling project. Many of you know that this has been funded by now, the base project. It has been 50% of the costs are taken by ICDP. And we have 10 proponents from 10 countries in South Africa. Nick Bukes is leading the effort. And by now we have almost gotten our money together, and we think we could start drilling maybe in the middle or late next year. We have a really good team looking at 4,400 meters of core that we hope to acquire. And all this, of course, is an effort to contribute to the scientific knowledge about South Africa's most recent World Heritage Site, the Barberton mountain lands. So this is my name and my address. Thanks for listening, and I'm looking forward to your questions.
Christoph, thank you for that fascinating talk. If you have any questions, raise your hand, jump right in and ask them, or you can even type them into the chat.
I think
anybody?
No one is there anymore. This happens to my lectures all the time.
I have a question.
Okay, thanks, Tony.
I just wanted to say thank you very much. I'm tuning in from Barberton, so I've been going up there quite often during lockdown. And yeah, good talk. Thank you very much.
You're welcome, Megan.
Tony, we interrupted you.
Thanks. My question is regarding the relatively thick chert bands in the within the crinklies. And Christoph, you mentioned that the growth of biomat sort of colonists, or whatever you want to call them, the archaea that constituted the biomats, grew inside these blisters,
top down,
from top down, and they sagged, or they seemed to be affected by gravity and hung downwards.
Yep.
How, if they are photosynthetic, how do they grow in the dark?
They don't. That is, at night they rest, or rather they metabolize. They use their sugar and their proteins, whatever they build at night, to tide themselves over until the sun comes back up, just like plants do.
Okay, but if they are covered by a dome of sand, there would be no light in there.
Yeah, good question. First, Tony, we don't really know how they worked, yeah, but their morphology and their setting within the sand blisters and the location of the sand blisters within the tidal zone suggests, note my verb, it suggests that they may have been photosynthetic, that is dependent on the light. Secondly, the cover, we don't know when they were active. Maybe they were active when the cover was still very thin and the blister had a cover of 3 or 4 sand grain diameters on top of them. And of course, quartz is great for stopping UV radiation, but it's transparent and would leave the visible light necessary for photosynthesis pass. So living in a transparent or translucent cave would be just the ticket, I think. It would protect yourself from the physical aberration of the surf. It would protect yourself from the UV radiation, and it would still allow you to have the light.
Great. I mean, that's an exciting possibility, really is.
If you and I lived in deep water and were forced to live on land now, where would you do that? You would try to build yourself a shelter. You would not crawl out and live on the open plain, exposing yourself to the atmosphere and to the meteorite impacts and the volcanism and the UV radiation. So I think finding as one of the first lines of evidence for early life on land, a cave, makes perfect sense.
Thanks a lot.
This must be Chris.
Hi.
Chris and his dog.
Hi. Thanks, Christoph. Christoph, do you think these similar organisms could have survived into things like the Pretoria group?
How old is the Pretoria group?
2 billion years.
We don't know. I mean, life could have become extinct a couple of times and reinvented a couple of times. And with Barberton, the big disconnect is that the top of the Moody's group doesn't go anywhere, right? There's the top of the Moody's group has no erosional contact to overlying rocks. It is just sticking into the air. We know nothing about the history between the surface history between 3200 and the start of the Black Reef at Kaapse at 2900 or so.
Beautiful. Yeah, thank you.
You're welcome. More questions?
Yeah, I've got another one if there's nobody else jumping in. We know the Kaapvaal Craton as the earliest known proto-continent, which name suggests to us that there were no others that are known. And does that suggest that prior to the Greenstone Belt era, there was actually no tectonic plates shifting around, and that plate tectonics grew as a result of the rising granites occurring as a phenomenon right throughout the globe, eventually forming these rafts, continental rafts, and that in fact before the Greenstone Belt was formed, there was actually no plate tectonics, no continental movement at all.
Indeed. Yeah, because there were no continents. Continents are something very special. And Tony, we talked about this a couple of times. But maybe for the rest of you, we know of no other planet that has continents. We know of no other planet that has liquid water. We know of no other planet that has life. Is there a connection in this triangle? Is one the prerequisite for another? Are continents prerequisites for life because of weathering and tidal zones and shallow water and exposure to photosynthesis, the possibility to do photosynthesis provided by these rafts that we call continents? And these are all questions that we cannot answer at present because we have no comparison. Yeah, we need to find continents. We need to identify continents on these exoplanets, but and undoubtedly the exoplanetologists will get there sooner or later. But at the moment we don't know. So while I think Unverwacht, Fig Tree, and Moody's groups do not represent any deposits in the conventional plate tectonic sense, such as rift or oceanic accretionary margin or foreland basins or anything. They are all products of semi-stable and vertically dominated tectonics. When I look at the Unverwacht Anticline again, I mean, that slide of me just at the almost at the very end, it must mean something to stick an oceanic plateau the size of Hawaii vertically into the ground. 10 kilometer stratigraphic thickness, tightly folded and plunging 90 degrees.
Thanks a lot.
Wow, when you put it that way. Bruce, you have a question?
Thank you. Thanks, Christoph. It's Bruce Rubich. I work on much later forms of life, reptiles and things like that I'm used to walking around on land. But I was very fascinated by your talk, and thank you for a really exciting development in the origins of life.
You're welcome.
I didn't quite understand the stalactite type situation, the things forming in caves. Is that in you were talking about it when you were talking about the intertidal? Do you see those caves as being in the intertidal zone?
Yes, and you have to imagine
that I've got a very bad connection.
Yeah, you have to visualize each of these black chert bands as the fill of a cave about 10 centimeters long and maybe maximum 1 centimeter high. They were blisters, sand blisters, and the roof of these caves was kept together by desiccating microbial mats. Should I expand?
Okay, thank you. So these sorry, my connection disappeared at that point. So you see these as little sort of blisters that happened and was about 1 centimeter high.
That's right. And then they were getting filled. They were probably first getting filled with water and kerogen, right, while they were active, kind of a soupy kind of kerogen. And when water withdrew at low tide, the kerogen started growing downward into the air-filled cavity. And then a few hours later, the cavity got filled again with water. And when metabolism finally ran out, they probably first calcified because we see remnant palisade carbonate. And that carbonate was then rather soon replaced by silica, stable along with all the silica cement in the surrounding sandstone, stabilizing the rock.
Thank you. Now, just as a point of interest, also, just it's more from an interest point of view to know something about the evidence of life in South Africa. Is the Unverwacht life, evidence of life, is it eukaryotic? And secondly, is that the earliest eukaryotic life in the world?
First half of the question, no, it's not. At least no one has ever seen a nucleus in one of these single cells. And the oldest known, or let's say generally accepted by the majority of reasonable scientists, occurrence of eukaryotic life, to my knowledge, comes from the gunflint chert, which is, I think,
Okay.
1.8, 1.9, 2.0 billion years old.
Okay, thank you. Thank you.
You're welcome.
And so is Onverwacht some of the earliest evidence of life on Earth?
Yes. Yeah. So I
Okay.
and in the publications of the last few years, people have stepped down through the stratigraphic hierarchy into lower and lower levels of the Onverwacht group. That is, they were approaching the core of the Onverwacht anticline by doing that, and therefore getting into these metamorphic temperatures that Tinus Klote already identified in the late 80s, early 90s as having been around 450, 500, 530 degrees Celsius. So the lowest sedimentary unit in the Greenstone Belt capable of preserving kerogen is the middle marker, separating the top of the Komati Formation base of the Hugenok. And that is 3472 million years old, I believe. And people have tried to find evidence of life, but so far, I believe they have not found any morphological fossils, but only chemofossils, that is kerogenous material depleted in 13C, depleted in the heavy carbon.
Thank you very much. Thank you.
Do we have any other questions for Christoph? Okay. Thank you so much for joining us. That was absolutely fascinating. Hope to see you in Barberton really soon.
Yes, I wish I'd be there.
We're looking forward to it. Everybody else, thank you so much for joining us, and we'll see you next week. Have a lovely weekend. Thanks, everybody.
Thank you for inviting me. Bye-bye.
Bye. Craig, if you could end the meeting for us, please.